It is my great pleasure to introduce to you all today's first speaker, Dr. Daniele Caprioli, a leading expert in drug addiction and relapse. Dr. Caprioli is an associate professor at Sapienza University of Rome, holding degrees in psychology and pharmacology. His research focuses on the neural and behavioral mechanisms of drug relapse. And today he will be sharing insights from his research on how specific sensory experiences, particularly those related to social interaction, may influence drug-seeking behavior. This work offers new perspectives on how environmental factors can affect addiction. So before we move on, if you have any questions during or after the presentation, you can write them on the query section below, and we will have around 5 to 10 minutes to answer your questions. Please join me in welcoming Dr. Daniele Caprioli.
Thank you very much, Nazahan and Helena. Hope the pronunciation is okay. Thank you very much for having me here. And it's a great pleasure because, this neurogon is very intriguing. It's managed to attract excellent speakers, which is great for the community. So today, as you just said, I will tell you something about, to an extent, my journey as a postdoc. Just let me see, there's something, okay, as a postdoc in Yavin Shaham lab. Actually, it will be a speaker tomorrow at 11 a.m. Bucharest time. And these are, of course, all my studies, etc. There is lots of people that I need to acknowledge. Certainly, Yavin has been inspirational. And also Marco Veniro, that at the time when I was a PhD student a postdoc, sorry, at NIDA, I met this guy, Marco. We are now very close friends, first, and collaborators, second. And we started this journey on this voluntary abstinence. And I will tell you something. I'm sure that Yavin tomorrow will provide you with more in-depth discussion on this subject. Now Marco is assistant professor at University of Maryland, so he had an amazing career, very fast. And also, these are the other collaborators I'm in depth with, along with the guys of my lab. These are the PhD students. Most of the data that I will present today have been collected by my two electrophysiologists, Claudio Marchetti and Ingrid Reverte Soler. So let's start. So with a very basic concept, what is addiction? So we know there's a classic definition that is written in essentially whole papers at the beginning in the introduction. Is a neurobiological disease characterized by compulsive drug use despite negative consequences. So this is a very established concept, okay? But of course, this definition can nicely integrate another perspective, the philosophical definition, which is a complex and neuropsychosocial a psychosocial puzzle that is featured by psychiatric comorbidity, limited socioeconomic opportunities, temporary myopic decision-making, denial, and the self-identity. This is an important concept because people going through life transition, such as, the loss of a home or family ties, and mental illnesses in general, suffer of a loss of identities. And the change involves a sense of loss of continuity, confusions about one's social role. And in a similar vein, addiction represents an identity loss. And that overcoming such loss requires some form of identity reconstruction. Some addicts identify themselves as addicts, and the self-identification can explain in part what we have just said before, why they use drugs and why they are entrapped in this sort of vicious circle. So long story short, for these individuals, recovery requires forming a new identity involving study, work, or family roles to replace their former substance-using identity. And this is essentially the root of our investigation, because the goals of addiction is stopping or reducing drug use while promoting a reallocation of behavior. So as a preclinical scientist, the first question that we asked was how to achieve suppression of drug-taking behavior by a non-drug reward, because this seems to be the core of what we should do. And as usual, the answer is typically in old-fashioned papers. And this is a beautiful paper from Sprague in 1940. He kind of revealed if we ask to a chimpanzee to choose between morphine or food, it will typically select an alternative incentive unless it's in acute withdrawal. But of course, the main inspiration actually comes, at least for me, came from Serge Ahmed, is one of his seminal studies, where he proved that if we give rats with an history of heroin, methamphetamine, cocaine, self-administration, the option to choose something alternative, an alternative incentive, in this case was a sucrose solution, rats will go for the alternative incentive. So what we have done is first to replicate these findings. Can we manage to induce a short voluntary abstinence in rats by presenting alternative incentives? And why this is particularly important? This is particularly important because the contingency management treatment in humans works to an extent exactly in the same way. In exchange of clean urine, for example, you might be rewarded with an alternative incentive, a voucher or something. And we know that this strategy promotes abstinence. And so we see this analogy. You might ask also, can we use a non-drug reward other than food in preclinical choice procedure? And this is what Marco has started with me when we were at NIDA. And again, some of the first inspiration are coming from the past. Chimpanzees have a profound social animals. And so we started to, well, if we build the boxes, for example, where we let animal with, again, choose an history of methamphetamine, for example. And in this box, there is also an adjacent room with a peer. Can we convince him to choose the alternative incentive, the social? And I will present you data that support this. And of course, these are publications that, they came out, the first one in 2018. We have now, we know that this is possible. And I will show you the data in a second. And we started the mechanism underlying these aspects. So let's transfer this logic to tackle the clinical problem, which is the high relapse rate during abstinence. You can see this nicely in this curve. The percentage of people that abstain over time dramatically drop. And this was nicely described in two independent papers very far apart in time. So how do we study this relapse? Well, this is for the one of you that are not familiar with these strategies. I will just give you a brief overview. Well, we first implant rats with a catheter in the jugular vein, and then we put in these operant boxes where we train them to depress the lever in order to obtain a bolus of whatever is your drug of choice. This presentation of the drug, this bolus, is typically associated with the cue. There is associated sort of a trigger for relapse. Then what you typically do is to test the animal in extinction condition. Why in extinction condition? Because typically when you want to do the neurobiology of things, you don't want the pharmacological compound in your preparation. And so what we are assessing how abstinence shape the craving, shaping this aberrant motivation. Of course, this procedure has some limitation. Why? Because the abstinence is typically experimentally imposed. What does it mean? It means that the experimenter remove the animal from the self-administration box and transfer back into the animal facility. And of course, in humans, something similar happen, of course, if you go to jail or if you are institutionalized. But the large majority of people is struggling to voluntarily abstain because of what? Well, because they are at risk of losing the job, for example, families, etc. There are many consequences associated. So there are some limitations with this strategy. And just a few words about craving. Craving is a complex construct, but something that we know, starting from the first observation of Gowan and Kleber in 1986, there is a dynamic, okay? And typically here, what you are observing is that the craving in rats increase over time, and the shape is a sort of an inverted U shape. So animal immediately the day after, just to put in other words, if you test their motivation to seek the drug, they typically have a relatively low motivation. But after many days, you see this pattern. And this pattern has been demonstrated in animal model with essentially all drugs of abuse. And there are now evidence also in humans, which makes the procedure per se robust. And we know that craving is one of the factors that can precipitate relapse. So what we have done is to revise the behavioral procedure. So this is, as I told you, has some limitation. There is forced abstinence. What we have done is to replace with, we call it voluntary abstinence. And here we set two strategies. A voluntary abstinence that is induced by palatable food pellets. In animal, this is important, that are not food deprived, okay? We are not playing with motivations in general. Or with a voluntary abstinence that is induced by a social peer that we named as a social-based voluntary abstinence. So let's have a look if these two procedures can sustain abstinence. And here are the data. So how does it work? Self-administration or food self-administration. Then we train the animal for meth, but now we have data also with heroin and with cocaine. And then this is how it looks like the voluntary abstinence. When you let them choose in 15 discrete trials between food or the social peer, what you are typically observing is food or the social alternative incentive is able to suppress entirely the methamphetamine seeking and taking. But then there's an important question. What is happening? So these two models, if we make a parallel based on what we said before, are models that are mirror in a way, for the case of food, the contingency management. And in the context of social self-administration, what is termed the social community approaches that also in this case was quite robustly in sustaining abstinence in humans. But what about craving? And here we found something that is kind of intriguing. With the food, craving incubates. So despite here, as I told you, food can suppress methamphetamine seeking and taking. When we discontinue the alternative incentive, food, the animal prepotently express high seeking, craving. You might say that is craving. We don't know. But with social, this is not happening. So it seems per se that social is able to counteract this seeking, this craving, this incubated craving phenomenon. We have started a characterization of the mechanism, and here we produce these two papers. We observed that the dorsomedial striatum neuronal ensemble play a role in the central amygdala pathway is implicated in this phenomenon. But I will not talk about this, but I will rather talk about what we are doing, one of the projects that we have here in Rome on the role of focusing the attention on the nucleus accumbens. Well, of course, this is, you all know that both clinical and preclinical evidence show that cue-induced drug seeking is largely associated with alterations in the neuronal activity and the plasticity within this important hub. And so we decided to focus our attention there. So this is data that are already published. We first wonder if the nucleus accumbens play a role in this incubated craving after food-based voluntary abstinence. So these are the behavioral data. Of course, we observe incubation. And something that we observe is an interesting anatomical dissociation. The nucleus core was activated. We revealed this by using FOS as a marker of neuronal activity. But this phenomenon, so you see this incubation, there is more FOS expression, but this phenomenon was not present in the nucleus accumbens shell. We also qualified the phenotype of the neurons. There are FOS positive by using RNA-scope and situ hybridization. And what we saw is that the large majority of the neurons are D1 positive. The nucleus accumbens core, the nucleus accumbens in general, we know there are medium spine neurons, and there are these two classes, the D1 and D2 subgroups. What we saw is that FOS positive neurons are largely D1, okay? Again, in the shell, there is not much going on. But something that is interesting here also, the D2 were implicated. Of course, the data that I showed you before are just correlational. So we move to causal approaches to test if the nucleus accumbens core was causally implicated in this cue-induced craving after food-based voluntary abstinence. We use as a first strategy muscarinic baclofen, which are GABA A, GABA B agonists. And what you see is a beautiful attenuation of cue-induced craving. This phenomenon will be not in the shell, supporting what we have just said, where the correlational data, the nucleus accumbens shell was not FOS positive, likely suggesting it is not involved in this phenomenon. And this causal manipulation explains this. What we have done then is, as I told you, the FOS positive neurons were D1 and D2 positive. So what we have done is to use raclopride, which is a D2 antagonist, a selective of sharing 39166. And what we observe in both the manipulation we observe in the nucleus accumbens core an attenuation of craving. We also use flupentixol is a D1, D2 antagonist. And again, what we observe is a similar attenuation. So suggesting that this D1 and D2, there is a coordinated action that play an important role in this form of relapse. We, of course, use alternative control. Why? Well, because in our animal model, there is a sort of built-in condition. These animals are also trained for sucrose self-administration, and we reproduce the same manipulation with masquemol baclofen, but we did not find any effect. Again, suggesting that the core, more than the shell, is implicated in this form of relapse. So I want to sum up this first block of findings before moving to the social-based voluntary abstinence. So our model can be used to study incubation of drug craving after prolonged voluntary abstinence, mimicking the human condition of relapse after successful contingency management treatment. Incubated med-seeking after food-based voluntary abstinence was associated with selective increase of FOS expression in the NAC core, but not shell. And FOS was co-labeled with both D1 and D2 MSNs. And the NAC core, but no shell injection of the GABA-A, GABA-B agonist, or the D1 and D2, or D1 selective and D2 selective dopaminergic antagonist reduce methamphetamine seeking at late abstinent time. So let's move now to the social reward as an alternative to drug use. I'm going to tell you why also we went in that direction, because some reviewers started to ask when we were publishing the mechanistic, they started to inquire, well, but with food, you might be at risk of inducing a sort of another addiction, food addiction, assuming that this is a valid concept. And so you might make things more complicated, and to an extent, this might not be translationally relevant. And for these reasons and other reasons, we decided, well, let's move in the social. The social is ecologically valid. It's extremely important for humans. Clinicians know this aspect very well. Okay, they know this since forever, essentially. That social is a critical factor. And so what we have done is build these boxes. Here there is let me tell you something about these boxes while the video is running. Here there is one lever with drug, and here is the lever. Then when depressed is opening automatically a door, and as you can see, there is a body there. And what they do is to start playing. You will see in the next trial that, look at this now. So here, the lever for the drug, in this case was cocaine, will come out, and the rat will be just in front of it. But you see, the lever pops out, but he ignore it. He turn on the social lever, and again, he depress the lever. This is truly wrong that we certainly observed for psychostimulants, at least the one that we have tested right now, which are cocaine and methamphetamine. With opioids, the story is different. And we now just put on bioarchive some of the findings with opioids. If you're interested, you can go there. The story is much more complicated with opioids. So Marco started his mechanistic understanding, mostly focusing on the central amygdala pathway. And he has revealed an important mechanism, a switch in the microcircuitry involving the interneuron PKC delta positive and somatostatin positive within the central amygdala. They are regulating this phenomenon. But I will not talk about this. But again, as I told you, our talk will be mostly revolving around the nucleus accumbens. So now here we have something. Let me sum up again. With food, we have incubated craving, but with social, we don't. okay? So, and this is also associated with a negligible FOS expression in the nucleus accumbens. So we decided, therefore, to focus our attention by using electrophysiology on the nucleus accumbens. What we know about the nucleus accumbens, we know actually lots of things during incubation of craving after the forced abstinence. Something that we know is that there's this mechanism, a calcium permeable AMPA receptor, progressively accumulates in medium spiny neurons of the nucleus accumbens. These are seminal studies from Marina Wolf. And she showed that this accumulation in the NAC, in particular the NAC core, underlying what are related to the incubated craving after forced abstinence. And this was true both for cocaine and methamphetamine. These are the two seminal studies. This is a Nature paper. In both cases is, you see, this is the first evidence of the GLUR2 lacking AMPA receptor, better termed. And these are the evidence with cocaine, and these are more recent evidence with methamphetamine. So based on this, we wondered, because this is a very well-established mechanism underlying incubated craving. Well, based on this, we wondered whether the social-based voluntary abstinence was somehow able, we don't know how, that was our hypothesis, to reverse this maladaptive plasticity by preventing calcium permeable AMPA accumulation within the nucleus accumbens, and by doing so, decreasing the incubation of craving. So we went in that direction. So let me tell you again briefly the experimental design. We trained the animal for social self-administration first, therefore methamphetamine self-administration. We had that here, because this is an electrophysiological experiment, a saline control. Then we tested the animal at an early abstinence phase. We interposed the voluntary abstinence in one group, and we contrast with the forced abstinence in another group. And then we tested the animal in a late abstinence time. So again, we have three experimental groups: saline, met, and forced. Saline, met, forced, and social voluntary. And saline and met self-administration, an early phase. Let's have a look at the data. So these are the data. Met social voluntary abstinence, and then methamphetamine self-administration. You see the animal acquire, they escalate their intake. This is the voluntary abstinence once again. And here, what we observe is relative to the forced abstinence condition, we observe this attenuation of craving. This is, to me, we were excited. This is the first time we reproduce this type of experiment in Italy. The previous experiment, at least the social concerning, were performed in the United States. So it's always great to replicate your own findings elsewhere. Exactly the same finding. We observe attenuation, socially induced. And then the first things that we have done is assessing the levels of AMPA subunit in forced and social-based voluntary abstinence. Why? Well, because we know that calcium permeable AMPA receptors are homomeric GLUA1 or heteromeric GLUA1, GLUA3. So what we have done is we use a BS3 cross-linking approach and assess the three subunit GLUA1, GLUA2, GLUA3. Now let's have a look at the data. This is what we found. Essentially nothing. We found that no differences across groups as far as concerned the GLUE1 subunit, as far as concerned the GLUE2 subunit, and as far as concerned the GLUE3 subunit. Nothing. So we found something peculiar. We finalized this experiment, and to an extent, we were scooped, at least in part, by Marina Wolf. She found exactly our finding by using Western blot. So we decided, well, we need to move to a more sensitive strategy, a more sensitive approach. And so we then decided to record, so the electrophysiology, of course, we decided to record evoked excitatory postsynaptic currents in medium spinal neurons of the NAC core, elicited by local stimulation in our three groups: saline, meth forced abstinence, and meth social voluntary abstinence. We know that CP-AMPA receptors are blocked by polyamines and at positive potential, and thus in their presence, currents and negative and positive potential are not symmetrical. We isolated the AMPA currents by adding the NMDA inhibitor, AP5, and GABA receptor inhibitor, picrotoxin to the bath. And here what we found, as you can see here, is this rectification. But this rectification was observed both in the forced abstinence group and the social abstinence group. This is kind of, again, puzzling. Why? Well, because in the forced abstinence group, we know that CP-AMPA are associated with incubated craving. But now here we observe the CP-AMPA accumulated, but craving is not there. So this was a kind of, peculiar. And I'm not an electrophysiologist, and I asked to my student, well, let's use a pharmacological strategy. And the strategy we use is a bath application of an aspam, which is a calcium permeable AMPA, selective calcium permeable AMPA receptor antagonist. And again, what we observe is the proof that in both groups, despite the behavioral dissociation, there is the presence of calcium permeable AMPA receptors. So, long story short, our first hypothesis failed. The social voluntary abstinence reduces incubation of meth craving. This is okay. But the rectification index is increasing both forced and social voluntary abstinence, which is not what we hypothesized. And this is both if you use a pure electrophysiological strategies or an electrophysiological and pharmacological strategy. Indeed, the application of the antagonist reduces AMPA EPSC amplitude in both forced and social voluntary abstinence routes. So we started to think, okay, we need to be more general. We need to look at different strategies. And we hypothesized a second scenario. Maybe the social-based voluntary abstinence manage to reduce the meth craving by increasing what? The inhibitory drive onto the medium spinal neurons. So this is what we wanted to, what we hope that was one of the mechanisms. And we set up a study for this. We measure the spontaneous inhibitory postsynaptic currents at late abstinence. And what we found, here you can see the cumulative frequency distribution curve for meth, forced, meth, social, and the saline control groups. But what you can see here is an indicative of an increase of the number of short interevent interval. But this was happening both in the forced and the social. Again, we did not find any dissociation with the saline group. So again, well, we also look at this where the frequency, but this are the amplitudes. Here we observe only a subtle effect. We are actually adding data to these curves on the amplitude of inhibitory current. It is evident cumulative curve, but not when looking at the mean, for example, which suggests it might not be such a robust phenomenon. And again, more importantly, if something, this effect is similar in both conditions. And we are searching for a dissociation at this level. So we also recorded spontaneous excitatory postsynaptic currents from the same cells. The main finding here is that the amplitude of these currents is reduced in forced rats, and this effect is prevented the first time by social-based voluntary abstinence. In other words, we found what we were looking for, a dissociation, a starting point in the attempt to understand the protective role of social over craving, incubated craving. We also recorded the spontaneous excitatory postsynaptic currents. We look into the frequency. We did not find much also in this case. So when we explore further the kinetics of these excitatory currents, we did not observe major differences in kinetics, either the rise or decay time of spontaneous excitatory postsynaptic currents. Therefore, the area under the curve difference that we saw before likely reflects the reduced amplitude that, as I told you, I just showed you in the previous slide. So to sum up here, met-exposed rats show increased frequency and amplitude of spontaneous inhibitory postsynaptic currents and late abstinence. The social-based voluntary abstinence does not prevent the met-induced increase in the inhibitory drive, and the forced abstinence from met was associated with a reduced amplitude and frequency of spontaneous excitatory postsynaptic currents. The social-based voluntary abstinence prevents changes in spontaneous EPSC occurring during forced abstinence from met, and met exposure per se increases inhibitory drive onto median spinal neurons regardless of the abstinence conditions. So to sum up, the voluntary social interaction prevents alteration in the excitatory neurotransmission occurring during met abstinence. So we started to have a handle to deploy our further experiment. We started to dig into this aspect, and we then focus on excitatory neurotransmission, and we decided to measure evoked rather than spontaneous current to investigate whether the origin of this alteration were pre or post-synaptic. So trying to tackle in detail, zoom in into this phenomenon. As you might remember, we observed first the reduced amplitude of spontaneous excitatory current. First. So quite surprisingly, when we moved to the evoked, when we stimulated at increasing intensities, we observe a robust increase of evoked glutamate currents in the forced abstinence rats. Here, this is, I think, one of our stronger effects. And there is a kind of a gradient between saline, social, voluntary abstinence, and the forced abstinence. Of course, this is to an extent in contrast with what we observe in the spontaneous excitatory, suggesting different changes at multiple logic. That's one of the reasons why we should look into the circuitry of this complexity. We then measure the paired pulse ratio to assess changes in the synaptic, in the presynaptic glutamate release. Consistent with the previous works by Funk and others, we observe a depression in the second synaptic response, indicating that glutamatergic input, so the presynaptic side, onto medium spiny neurons display a high probability of release. We did not observe differences across groups. We finally measured the AMPA-NMDA ratio and found an increased ratio only in the first group, suggesting that the increased amplitudes in evoked currents are the result of a higher number of AMPA receptors. So to sum up this complex set of data, trying to make sense of all of this. So what we can, describe this phenomenon different level, convergent and divergent. So the convergent effect of the meth-forced versus meth-social abstinence. So in other words, incubated versus non-incubated craving are associated with an increased spontaneous inhibitory postsynaptic frequency and amplitude and calcium permeable AMPA accumulation. But there are also divergent effects in between these two groups. And now we are exploring further this, that there is a decrease of spontaneous EPSC activity in the forced abstinence group relative to the social group and the saline group. And there is an increased AMPA-NMDA ratio in the forced abstinence group relative to the voluntary. So, long story short, if we need to take home message of all this data, the attenuation of glutamatergic plasticity alteration in the nucleus accumbens likely underpins the reduced methamphetamine craving observed during the prolonged abstinence. We are now exploring a putative target of this complex that might have shaped this. And of course, an obvious target is oxytocin, but we're working on it, but I don't have robust evidence to show you about it. So in conclusion, now I want to make a different type of conclusions, more broad conclusions, okay, for clinicians or students in general. So the first one is that our model can be used to study drug seeking and therefore craving induced by loss of an alternative drug reward after prolonged voluntary abstinence. As I told you, these are mimicking and contingency management, or we believe so, and the community reinforcement approach. As far as social is concerned, understanding how social factors modulate drug use is important to improve strategy to prevent relapse to drug use. The critical features of human addiction, we believe, should be incorporated into complementary animal models. We believe that this is an important aspect. Why? Well, because it's important, for example, to pharmacologists to develop environmetics, development of drugs that mimic positive environmental effects. And we know that social interaction, hygienic social interaction, the social domain is extremely complex, of course. But if we can understand, we can harness, we can tackle which are the mechanism that social interaction put in place to counteract the craving, we might develop environmetics that leverage these aspects. And as argued by Marco and Yavin on the concept of reverse translation, mimic successful treatment in humans in animal models of drug addiction. Why this is also particularly relevant? Well, because we know, as far as concerned, the pharmacological strategies, we are, despite many years of research, we are kind of behind, or at least this is the impression that we got. So we test many compounds, but typically do not translate. So we might go back, for example, to explore better or inquire better the animal models that we are using and where we are testing our pharmacological strategies. And that's it. And thank you again, Najan and Elena, for your kind invitation. And now I'm open to questions.
Thank you, Professor, for this great presentation. As our professor also mentioned, you can ask your questions in the Q&A section below, as we stated. While we wait for your questions, I have one question. Based on these rodent findings, how could we design social-based interventions for human addiction treatment? Do you work on these?
Yeah, well, we actually, well, this is actually something very well established. There are so-called social community approaches where we have in Rome, I'm actually running a clinical trials in this context. So essentially, there are communities with the psychologists, the doctor, etc., where people that suffer of substance use disorder goes there, and they are in a sort of a protective context where they work on rebuild the social interaction. One of the complexity, of course, of the typically induced relapse is when these people go back home in contexts that are likely associated with this. But as far as I can tell you, this social community approaches are extremely important. Okay, they also asked you to start your video. Oh, sorry, there is a window that pop up. Okay, so this is something that we already have, and we know that works. And what we're harnessing is trying to understand how it works, which are the mechanism that we can leverage. And I can tell you that oxytocin, of course, it might not be a surprise. It might be something that we can harness. And of course, there are also clinical trials on the impact of the oxytocin, for example, in preventing or attenuating craving. And social, we still do not have solid data on this aspect, but the social interaction by, for example, promoting the activation of the paraventricular nucleus of the hypothalamus on the nucleus accumbens, we know that there are strong projection there, and releasing oxytocin might shape the plasticity in this brain region. And we have also preclinical evidence that suggests this. There are beautiful studies from Malenka on the social interaction, suggesting that oxytocin, for example, via a complex interplay with serotonin, frankly, it's able to shape the plasticity within the nucleus accumbens. So we are kind of tackling this this mechanism, and we'll see if the data will support our hypothesis. But as you can see, as you saw, well, our three hypotheses, two failed, unfortunately. So it's never easy.
I see. Thank you, Professor. I'm looking for questions, but it seems we don't have any question yet from our audience.
Well, maybe it was too much electrophysiology. Those data are typically a bit hard to digest, at least for students.
Yeah, maybe. I also want to ask, during these experiments, did you perceive any resilience to social isolation from the rats?
Sorry, can you come again? No, I didn't get it.
Were there any resilience to social isolation to others from the rats during these experiments? Did you receive any findings?
Well, social isolation, the forced abstinence condition is a social, essentially all the animals in the forced abstinence condition, which would be, humans are going to jail, or they typically express a very high craving. So, well, as far as concerned our animals, yes, our animals are socially isolated, but Marco did also some experiment with a socially enriched environment, and in both occasions, in both cases, the craving goes down. So it's not the housing condition per se that affect the craving, the attenuation of craving.
I see. We have a question. They are asking if the diet and food in abstinence can contribute to craving.
Well, this was I don't have an answer to this, but it's likely that might be an interaction between the two conditions. Although what I can tell you is that our animals are never food deprived, so it's unlikely that this is. And also we got data both with, of course, sucrose, but also with saccharin, which doesn't have any caloric content. So, but of course it might, and this is one of the reasons why we moved to a more ecologically valid platform.
Thank you, Professor. If there are no other questions,